I'm going to tell you a little story about Zeiss imaging, volumetric imaging, and maybe let's start with a little quiz. So my colleague Radu is also here representing Zeiss Romania, and he's offering €5 for the person who responds correctly. No, I'm just kidding. So first of all, have you heard about Zeiss? You heard about Zeiss?
Yes.
You heard. So that means you use Zeiss, right?
Yes.
What can you tell me? So Zeiss, what is the first thing that comes into your mind when you say Zeiss?
Precision.
Precision, that's true. And of course, microscopy. And actually, Zeiss was the first commercial microscopy, let's say company, although we are a foundation, so, but okay, we can say company like this. But the first little quiz, can you guess more or less around which year actually we started as a company? What we started, they started as a company. Don't be shy. 2000, 1900s. 1900s. It was actually before that, 1846. I don't think it's mentioned here, but 1846 actually we have our main founding father, Carl Zeiss. That's why we have the name Zeiss. And then he associated with a very famous physicist that I'm sure you know, Carl Abbe. He's the one that has discovered, let's say, this equation that defines the maximum resolution you can achieve with a microscope. And then later on also he associated with, I think he was a chemist, Dr. Otto. And Dr. Otto actually had started a very nice factory for glass. And I'm sure that you're using maybe in your lab, you're using glassware from Otto, because the factory still exists today. So 1846. We are doing microscopy. Do you think we are doing also something else? Another little quiz for €5? We didn't have any winners so far, so this is real. So we do microscopy. What else are we doing? Who knows?
Glasses.
Glasses. So what you see here, my glass is actually from Carl Zeiss. Really very good glasses. What else? Two more things. Do we have any medical doctors here? We don't have medical doctors. Okay, then I will jump in. So, we're still doing microscopy, and microscopy sits under this industrial quality and research. So we're doing for the research microscopy solution, we are called for this part. We have the consumer market, you see with the binoculars, also with this lenses like this for the glasses. We're doing medical technology, and Radu, I think in Romania especially, there are a lot of size, let's call it machines, robots in the hospitals, right, for the surgeries and stuff like that. But believe it or not, our main business nowadays is still optics, but we are doing this highly precise optics for semiconductor technology, for lithography. I don't know if you heard about ASML, the giant actually from Netherlands, who are doing this kind of chips basically for the whole things. So we are providing them with the optics, with the right optics to do this part. Good, 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 good. Coming back to the microscopy, we have a very broad portfolio. As you can see it in here, which kind of microscopes do I identify? Very quick. €3, please.
Yeah, I think we deserve €10 for that.
€10 for that, right? So we're having the portfolio for light microscopy, the classical photonical, let's call it microscopy. We are also doing what? Electron microscopy. And in the middle, you'll see also, this is also a microscope for X-ray. So we're also doing X-ray microscopy. Why do we show it like this? Well, first of all, to show off. No, I'm just joking. Maybe also to show, to say that, look, we have a very broad portfolio, but are actually living the days of the correlative microscopy. So we're trying to correlate everything with everything, kind of, right? We see here correlative microscopy all across light microscopy, electron, and X-ray machines. What does it mean, correlative microscopy, in a nutshell? You have your sample, you take it to a light microscope, take an image, bring it to an electron microscope, and you put the images together. Let me show you an example. This is from light microscopy to light microscopy. This is a sample with a mouse brain slice. First, we took it to a slide scanner. We took the overview image, and then we went to a confocal, and we took the, let's say, higher resolution images. And then we can put it together, and you're going to see a very nice movie, starting with an image from your phone, like Catherine is doing right now. He's recording. So you can start with that, and then you can just zoom in, and maybe if you're a PhD, if you're a postdoc or a student, you can show to your PI, Look, professor, what I have done with the sample. So everything comes very nicely into the right context. Good. And now we're coming to the subject of the day, volumetric imaging. What does it mean? Why would it be useful? Who can tell me that? When you think about volumetric imaging.
For 3D mapping of features inside.
3D mapping. So
computer objects.
we're living actually the days that we see more and more in research. People are moving, first of all, from, let's say, classical culture cells, as you can see it in here. They are moving to more and more, let's say, tissue, organoids, spheroids. I heard probably you all know about this kind of things. So we're having much more complex samples in 3D, just to put it like that. So then if you use the classical approaches, you're going to get, for example, a confocal microscope. You can get a very nice image, right? But it will take forever to get the whole thing as a volume, right? So that's why we need to have this kind of things. But also, if you think about the culture cells, or even the tissue that can be alive, at the end of the day, everything is in 3D, smaller 3D or larger 3D, right? So we need these volumetric images to cover large specimens, but also to be very gentle, right? If you're using a microscope and you put too much light, what will happen with your sample? Who can tell me that? Photo bleaching, as you can see here, phototoxicity, a lot of bad things in a way for your sample. So then you have the question, your results that you see are really relevant or not? If you see a lot of photo bleaching, if you see your cells are dying, that's not really good for your experiments, right? Good, and this is a very nice, maybe a bit dramatic comparison for the gentleness of imaging. So imagine that you are in a very nice beach. I would like to be there actually right now, to be honest. And you have just a little sun in the morning and you have a very nice breeze and it's super nice and you feel very relaxed. Imagine this is your sample under the microscope. Now imagine that you don't have one sun, but you have 10, 20 stars, which are shining a lot of light on you. How would you feel? Very stressed, right? So your sample will feel the same. That's why it's very important to do not only volumetric imaging, but also very, very gentle. Now, which kind of technologies can we say they are really volumetric? First of all, we have what we call Lightsheet microscopy portfolio. Have you heard about Lightsheet? You have heard about Lightsheet? Yeah. Have you used Lightsheet microscopy?
Yeah, tried it.
You tried it? Okay.
Demos, you made demos.
You made demos. Okay, cool. So what would be different from a Lightsheet microscope compared to a classical microscope? Let's say this is your specimen. In a classical, let's say confocal epifluorescence microscopy, the problem is that you put a lot of light on your sample. So you have the same path for the excitation and for the emission, going for the same object and then for the sample and stuff like that. And you see the red color. And the red color means it's bad, right? So that means that the specimen can be very, very much stressed. In the Lightsheet illumination, what we do, we decouple the two light paths. So we have excitation objectives that will create a sheet of light, therefore the name Lightsheet. And we have emission objectives which are perpendicular, orthogonal, basically collecting the light. And as a result, you just illuminate your specimen in a single plane and you move your specimen through the sheet. As a result, it's super fast and it's also super gentle to your sample. Two little monkeys we have in here, two microscopes for that. That was the first one, it's called Lightsheet 7. The one on the left-hand side was launched maybe 5-6 years ago, is the lattice Lightsheet. They're very complementary to each other and they cover a very nice broad range of applications going from cells, with the lattice Lightsheet, to large specimen. My favorite one, you see this one, I like to call it DJ Octopus. In Belgium, we have a lab using our Lightsheet and they made a full atlas of this certain octopus subspecies actually of the embryos and stuff like this. Good. Complementary lattice Lightsheet for smaller samples. The classical Lightsheet 7, called Gaussian Lightsheet, is for really, really large specimen. And the first few things I want to show, this is the classical one, the Gaussian Lightsheet. This we launched actually so we could do volumetric imaging since 2012 or 13, if I'm not mistaken. What can you do with it? Like I was telling you in the principle, so we have separated excitation objective, so we can illuminate from the left, we can do it from the right, and this is the emission objective. And then as you're going to see in the movie, we just move the sample very fast through the sheet of light and then we detect the fluorescence. Another very nice thing, with a little camera, you can see the perspective of the specimen and you can rotate it. So you can pre-position in the right position for the imaging that you'd like to have in the perspective and stuff like this. So 2012, I think it was launched for living creatures. So you could see fast dynamics like that. This is the beating heart of a zebrafish. You see labeled with 2 colors, 2 fluorophores simultaneous. 80 frames per second, so really super fast. You could see this very nice beating. It was also ideal for people that looks at development. This is this one, I think the Drosophila embryo developing, as you can see it in here. Plant people loved it, so they could also look, they could put plants in there, and they could see root growth or flower growth like this case over here. And I have another very nice example, also Drosophila embryo, so you can see the division followed with the light sheet of this. And then using that, you can also do post-processing, and you're gonna see it next. You can track actually single cells, how do they move, where do they go, and things like that. So this is the real volumetric imaging, right? For large specimens, living specimen, with the Lightsheet 7. Now, in 2014, there was a very interesting thing happening. What is the problem in microscopy? If you have very thick specimens like this one, you see here, this is a whole paw. This is a full brain from the mouse, and this is from a very adult, let's say, probably adult mouse or even a rat. What is the problem if you have the tissue like this in microscopy? Who can tell me? Scattering. And then you cannot really image deep in the tissue. Now, have a look at these things, and actually the whole slide, maybe you've heard of it. So nowadays, especially, these clearing methods, as I say in here, to make the specimen really transparent, they boom like crazy. And I remember in 2014, so I took, I think it was for scale, I put a brain like that, and I had to wait for like almost 2 months to see a bit of clearing. Now, the same kind of sample with things, especially derived from disco, it takes a couple of hours, maybe even less to clear it like that. And since you have it clear like this, then you can do very nice imaging. So we adapted our light sheet for this clearing methods, for different refractive indexes. Because, you know, every piece of tissue has a different clearing method, actually, that responds proper to that. So when we can image all of these things with different refractive indices. This is the volumetric for the big samples, for the Lightsheet 7. Its brother, Lattice Lightsheet 7, as you can see in here, it doesn't look like a real microscope, right? You don't see any binoculars, but it is a real microscope, a very, very, very nice one. Also very expensive, but totally worth it. And if you look in here, it's actually adapted for the cell cultures, for the like a regular inverted microscope. So you can do very fast imaging for live cell cultures. And I'm going to show you some super nice images. I hope you like it at least. Volumetric and very gentle. But before that, just to tell you that the optical components and the systems, they are crazily, highly engineering involved, just to put it like that. This stage, for example, is unique for this system. It's a 5-axis stage. So 2D, 3D, traveling large distances. It can also correct for the tilt. If you have a tilt of your sample, for example, of your dish. We have auto immersion on this and a lot of, sorry, you want to take a picture? Please. Shall I be in the picture now? Good. Have a look at this. This is one of my favorite. These are living cells with live acting with GFP, green fluorescent protein, volumetric, continuously for 9 hours, more than 1 million images. And look how happy they are. There is no photo bleaching, there is no phototoxicity, and they just spread and do their own stuff actually continuously. This is what we call really being gentle for imaging using a microscope. Another little video, dividing cells. You have seen dividing cells for sure in some other videos. But please have a look at this. You're going to see multiple divisions. That means a cell divides, and then you have a second round of division from the daughter cells. And this is also super nice because it means that your imaging system is really, really gentle. You will see in here probably. I don't know if it was already there. This is the daughter cell, and it has a second division. Next one, and this is the last one that I'm going to show for the lattice light sheet. Please have a look in here. And you're going to see at a certain moment a division. And you're going to say, okay, so what's so special on that? We have seen dividing cells before. I don't know if it's written in there, This is mitochondrial level, but these cells are really special. These are astrocytes. So from brain. And this is amazing because it's super duper rare to see astrocytes dividing under a microscope. And that brings the proof that this technology is indeed super, super gentle if such a cell line that is super sensitive really goes on and it makes division. I cannot give you details because we're still working for the marketing on that, but it was a really happy story behind it. Maybe not the time to say it right now. Good. So Lightsheet portfolio, lattice Lightsheet, Lightsheet 7. The last one that we actually have it also in here because we have a microscope in the other room. So if you have the time, please pass by. I can show you some stuff around it. Not around it, on it actually. It's associated with our confocal microscope, but it's not a confocal technology. It's called Lightfield 4D, and the name says it all. One snap, one volume. What you have seen so far with the Lightsheet 7, with the lattice Lightsheet, you still have to make a stack. You take your sample and goes through the Lightsheet, but it's still a stacking, right? With this one, you don't do a stack. You do a simple 2D image, and you end up with a volume. How does it work? You're going to see a little movie here. So you have an LED light source. You illuminate like you normally do with the epifluorescence microscope, and the whole magic thing happens here. In front of the camera, we have this area of lenses, 37 in number, and every single lens gets information from the focal plane, but also the ring ones actually get information from different depths and different angles. So at the end, from this kind of things, we get information about volumes, basically. So let me show you a very nice thing. It was for the first time where this customer has really seen instantaneous blood flow in a zebrafish. And it was really amazing because it looks totally different than what you would expect. You know, if you use some other technology, you still have to do a stack. And if you do a stack, the dynamics change. You're missing information. With this, I could really nicely see this kind of circular blood flow from the zebrafish development. I don't want to get too much into technical details, but just to show you the principle very quick, like I said, this is your sample, you illuminate, you collect information, you send it to the lenses, and like I was telling you, every single lens has a different perspective of your 3D arrangement of your sample. And from that, the software recreates for you the volume, which depending on the objective that you use, it will be smaller or larger or smaller. I'm going to show you some examples. You can do very fast imaging, and this time we're speaking about single color. 80 is the number, but it's not frames per second. We're speaking about volumes per second. So 80 volumes per second. This is a very nice one that shows you, for example, this is the beating heart of the zebrafish. And this is how you see it on the raw data, images collected with different lenses from different parts. And if we zoom in, you're going to see exactly what I was telling you. You see different perspectives of your sample, of your beating heart, like that. And with this in mind, let me show you some nice images as well. So this is taken with a 10 times objective, and this is a cardiac organoid. And what you're looking here is like basically beating, like a beating heart, right? And we have a calcium indicator, and this is for the first time that can look to the activity instantaneous in 3D of this beating heart. This is with the 10 times. This is another very nice one, spheroids. This spheroid is not alive, it's fixed, it's expanded, and the 10 times objective, we could capture the whole volume of 1.5 millimeter. So the acquisition for this one took like 1-2 seconds, just to acquire this thing with the 10 times objective. So we got the spheroid, but the spheroid was actually part of a multi-wall plate with many spheroids. So then you can do a screening like that. And what you see in here, it's a movie from the whole multi-wall plate. This whole image took up to 3 minutes with this volumetric imaging part, which is fantastic because if you look at the fluorophores, there are like 4 colors in there. So 4 colors, multiple spheroids within 3 minutes. It's a dream, kind of. Now, you should ask me, and I see you're very enthusiastic to ask me, why did you put it on a confocal microscope? We put it on a confocal microscope because we want to combine technology. So let me give you an example. The zebrafish, calcium imaging in the neurons. I start the movie, and you're going to see some bubbles. Again, with the Lightfield 4D, we capture in different planes basically instantaneously the calcium activity, the peaks basically. And after doing this with the light field, we're moving to confocal or super resolution, and we get the highly detailed information, and we just put it together. So we combine the different technologies. I let it play. I take a sip of water. If you have questions, you can also ask questions. Let's not make it very formal. You have questions so far?
What do you mean by bubbles?
Well, bubble is not the right word. It was emphasizing that different neurons basically from the zebrafish which are on different planes. So when I started the movie, when I say bubbles, it's to show, look, there is a neuron here, one on a different depth, one on a different depth, and thanks to the Lightfield technology, you can see them all in different depths actually, making these peaks, simultaneous peaks of calcium waving. This is the image then taken with the AR scan with our super resolution detector. And then you can combine it, so you get different kind of information from different perspectives. Good. I still have like 4-5 slides. That was the volumetric. Do you have questions on that? Other questions? Don't be shy. If not, I want to point just a few things about smart microscopy. Have you heard about smart microscopy?
We're building them.
Sorry?
We are building.
You are building up. Okay. Yeah, that's great. That's smart to have a smart microscopy. Well, smart microscopy is already here. But when you think about smart microscopy, what does it mean, right? Let me give you a few examples from my perspective what it does mean this smart microscopy. For example, at Zeiss, in our Zen software, we have, for example, in the Zen software, this kind of workflow which is called guided acquisition. It's a piece of software. I like to call it actually a sandwich type of experiment. What can you do with it? First of all, with the low magnification from your microscope, you do an overview. Then you have an image analysis script that is looking for certain processes, which you can define this image analysis script. And once the software detects those red processes, whatever you do in there, it switches to a higher magnification and is doing detailed imaging. Let me give you a nice example. You see here, these are living cells. So first you do an overview with a low magnification, and then you run this image analysis script, which is looking for dividing cells. In the moment when it's finding dividing cells, it's zooming on it and is doing a Z-stack, for example, in there and getting really the high resolution images out of that. This is an example of smart automated workflow, so smart microscopy, right? But then you can do more. We have what we call, Zen again, it's our software, stands for Zeiss Efficient Navigation, and we have what we call the API. API basically, how shall I describe it, Arthur? Application programming interface. It's a programming interface basically in the software that allows you to do kind of everything. It's a kind of sky is the limit. So you can do end-to-end workflows for image acquisition, for image processing, you name it. In the background, basically, it's Python programming, right? Am I correct in saying that?
Yeah.
We have programmers in here, so that's why I'm looking at them. Otherwise, I'm not with it. So with this kind of things that we have by default in our software, you can build your own experiment. You can implement also third-party things, like, to give you a very basic example, a microfluidic device or a third-party component from a different company. So that's why I'm saying with this kind of things, which is really smart microscopy nowadays, you can build your own very complex advanced workflow, end to end. And for that, to support you, we have a dedicated department which is called Solutions Labs. Programmers sitting 24/7, well, not really 24/7, 24/5, I would say, in Munich, in Germany. So they do customized solutions. So if you're coming, for example, and say, look, I want to have that solution, scripts and stuff like that, they make a case, they make a study, and then together with partners, and we have some partners here, Smart Labs, colleagues from Romania, they're developing a workflow for you, for your specific target. This is also smart microscopy. And I will give you, this is the last 2-3 examples. What does it mean also smart microscopy? For example, colleagues from Smart Labs, we had a case in Belgium where we had a confocal microscope, but then the customer wanted to put on top of it an atomic force microscope. So it's great, but it's 2 different technologies, 2 different softwares and stuff like that. So they did a very nice, let's call it synchronization using this API, Python, correct me if I'm wrong. So they can do, it's basically integration, so there is a very nice feedback given from the confocal to AFM or the other way around. So it's working very nicely as a workflow for the customer. But you can do more than that. For example, another thing is tracking for plants, but it can be zebrafish. It can be even cells. If they're moving out of your field of view, you can do this tracking macro, again done by the colleagues from Smart Labs, and then it's always looking for the same sample in time if you want to do it overnight or over 2 days and stuff like this. And this is for materials also. Image analysis can be part of the smart microscopy, because if you get 10,000 images, you don't want to really say do manual annotations, manual image analysis. You can also automate everything for the image processing, no matter if it is material microscopy, electron microscopy, biology, you name it. And I stop here. Thank you so much. I hope I didn't close the time too much. It's really half an hour. I don't know if you have questions.
Thank you for sponsoring us.
Radu. I'm poor.
And
Radu
also
is rich.
for being so sharp in keeping the time.
Thank you.
So yes, Professor Radulan.
Thanks for an impressive presentation. Do you have any images of Lightsheet microscopy which are not fluorescence?
Which are not fluorescence. You mean like a transmitted, a bright field, classical ones,
No. Still Lightsheet, but
Yeah.
not fluorescence.
without fluorescence? I don't have it in the presentation, but we do have it. So for plants, for example, we have also a lamp inside.
For roots, for instance. Was
For
cut
roots,
inside.
yeah, for roots
For
we
the
have the same
current
thing.
inject the fluorophore.
We have a workaround for that. So by default, it's not really, we don't have a bright field for light sheet, just to put it like that. But we do a treatment that we have a lamp that we trigger with a laser, so that we can do also this bright field imaging, and we associate it together with the fluorescence in our part. So you can do it. And if you want, sorry, what's your name?
Adrian.
Adrian. I will send you some examples of that from plants. In Czech Republic, we have two places, one in Olomouc and I think the second one in Brno, where they have really crazy workflow for plant imaging using our light sheet. They even have in one light sheet, they have a lamp that is triggered to simulate the daylight, so the root will grow like normally it will grow.
I will be interested in that. So
Okay, I will get back to you.
I'm here.
Okay, that's it. That's a real question. Other questions? No.
It's also to be noticed that the full equipment is just across the hall, and it will be a fitting out to take
Yes.
because it does have this 4D facility, which I think it's really exciting. And it's also possible to bring samples if the case, and this is also applicable to other instruments that are on display here.
So if you have any samples, I'm here until tomorrow at noon. Maybe short time, but I can do some imaging for you. Just bring it.
Super.
Yes.
I would like to ask if the AFM
Yeah.
device can be used also to measure molecular interactions, forces.
I mean, the FM it can be used for that.
No, your device is combined because in principle it's used for microscopy, but can be used also to measure molecular interactions.
Molecular interactions, yes, we can do it. So we have special technologies for that. If you want to say like protein interactions. So
Protein
Yes.
glycan.
yes, we have this kind of things. We have fluorescence correlation spectroscopy. We have things called dynamics profiler. I have a whole presentation on that, but if you're around, maybe I'll look for you. You're here, still here today and tomorrow. We have a bunch of technology that can look at that actually.
Thank you.
So even the system that we have it in here, I think it can do it. So this is the entry level confocal. I will look for you maybe in one of the breaks and I'll show you very quickly what we have on that. Yeah, but this is the name: fluorescence correlation, cross correlation spectroscopy, and we have also spectral RICS, raster image correlation spectroscopy. You can go up to 5 pairs to look if they're interacting 2 by 2. So quite a few.
Thank you very much.
Thank you.
Thank you very much.
Okay.
And once again, I extend the invitation to continue the discussion if the case across the hall.